All right, I think we left off here looking at the soil system and hopefully all the lovely nutrients, the minerals that are attached to the soil particles. So here you have a clay particle, and if it's clay or sand or silt or whatever, negatively charged surface of the soil particle, negatively charged. So that means the positively charged minerals can attach to that and bind to the soil. The negatively charged ones cannot. They will stay in solution and potentially percolate down into the groundwater, ground table. And that can be a problem with runoff from irrigation fields that are irrigated or fertilized too heavily with nitrogen. We'll talk a little bit about that. So here you're seeing the cation exchange idea, where you have cations on a soil particle getting exchanged by other cations coming along and bumping them off the soil particle, so to speak. And once they come off the soil particle, hopefully they're going to readily dissolve in the soil water, and then the roots can finally take up those minerals. Roots do release protons as part of their physiological processes, so that's why they're trying to show it's coming from the roots. So you certainly have that. And remember, with the lyotropic series, the one, the hydrogen it has a good binding affinity. So the fact that the roots are doing this is allowing them to, directly participate in the cation exchange process, where the hydrogen cations, the protons that they secrete, can be used in the cation exchange process. You're also seeing here that the roots are carrying out aerobic respiration, just like we are. We're giving off CO2 right now. Plants do very similar to what we do. And you can see that they're showing you when you have CO2 gas mixing with the soil water, you do end up with protons as a product. So again, a couple of different ways that you can see protons building up in the soil to allow for that cation exchange. Okay. Now, if you've taken the soils class or you're currently in it, you know that the soil composition is tremendously varied depending on what site you're looking at. This is just a tiny little snapshot just to try to get you to think about the idea of particle size primarily. So here we're looking at size of just 3 different types of clay particles. And you can see that just those within clay, you get a variety of diameters and shapes. And then the cation exchange capacity is your last row on this table. And the higher the number, the better that they are at that, okay? But there is a trade-off. Obviously, you can't have a soil where you want to grow plants that's all clay. It just won't work. So there's a definite trade-off. Yes, it has great cation exchange properties, but you don't want total clay. So you want mixture. So again, just looking at this idea of cation exchange, where you have cations that, if you're imagining either being released by the roots or just percolating through the soil with a rain event, that you can get this cation exchange process in the soil system. Here's another. Oh, and then here we're looking at the idea where the plant. So here they're trying to show how the plant, excuse me, is releasing the protons. So this is the plant root, and it's releasing the protons that can then do cation exchange. Okay, so they're just trying to show that mechanism. And here, let's see, here they're showing it how sometimes this is a sacrifice of energy to get this to happen. And so the plant root is going through primarily active transport here, where it's pumping protons out into the soil water system, and then the proton can participate in cation. So sometimes it is a sacrifice on this, in the sense of energy that the plant has to use to get those protons out and start doing the cation exchange. So that's all, the cation, but there's anions that the plant needs as well. And they can't bind to the soil particles. So there's not like this reservoir available for the plant to take advantage of. And so it's much more patchy in terms of when the plant will have access to these anions. And so this can, this explains, this is a big part of why growers fertilize their fields with nitrogen, because nitrogen isn't found in the soil system to very high levels. Most plants, when you think about plants, their favorite form of nitrogen is actually, the fertilizer gets applied as ammonia, but natural, they like nitrate. NO3 minus is nitrate. That's their favorite. Most plants, there's some exceptions, but I'm speaking most agricultural plants right now. And so, well, even non-ag plants, nitrate is preferred. And so that's negatively charged, that won't stick to the soil particle. So they have limited and patchy access to that. And so, oh, there it is, NO3 minus. So then it will, because it's not attaching to the soil particles, it'll just percolate through the soil system with the water. And if the roots don't grab it, it just keeps going. And so if you don't irrigate or fertilize your soil the right way, and this has been historically done for a long time, they put too much nitrogen fertilizer into the soil system, and then it washes down into the water table, and it creates pollution effects. And the fancy word is eutrophication. And so that just means that you have too much organic matter showing up, pollution into the runoff from ag fields and other pollution events. And it can cause a big, big problem in the lakes and rivers and streams and oceans. So eutrophication, that's where typically a great way to have that happen is fertilizer runoff from ag fields. That's a big culprit. And so it puts too much organic matter essentially into the water systems. And then you get these blooms, harmful algal blooms, you get cyanobacterial blooms. And really what ends up happening in the end is that these population explosions scarf up so much of the free oxygen in the water that now anything that needs oxygen starts to die. That's why you start to see fish just dying in big masses. It's because the microbes just pretty much scarfed up all the oxygen for themselves, and the fish are left with not enough oxygen in the water to breathe, and they start dying. So it's just like this domino impact. Here again, just trying to impress on you the idea that anything that's negatively charged, nitrate, sulfate, those cannot attach to the soil particles. Okay, so again, just a visual of what we're covering here. So they were able to figure that out in pretty short order. It's basic, soil chemistry that they were investigating. And then they started asking, what is the typical pH of soils? And a lot of this data is coming from ag fields, but they've done enough now in natural environments that it's safe to say that most areas have a pH that's slightly acidic. The soil is slightly acidic. There's obviously some exceptions to this, but by and large, pH 5.5 to 6.5. I do need you to know those exact numbers, 5.5 to 6.5. That's the range. And just remember now, when you calculate pH, it's the negative log of the proton concentration or the hydrogen ion concentration. So we're looking at how much H plus is present. And the more that you have that, the more acidic. And so when they started investigating the soil chemistry of pH 5.5 to 6.5, and they started looking at cation exchange and the minerals under those conditions, they do find that the minerals are more available to the plant under slightly acidic conditions, okay, just because of the soil chemistry of the cation exchange process. It really makes those minerals, once they get bumped off the soil particle, that slightly acidic makes the minerals go into solution that much easier, and so they're more soluble. The other thing about a slightly acidic soil is that fungi are very happy in acidic conditions. And so this is a good thing and a bad thing for plants, because there's plenty of fungi that are very beneficial and absolutely crucial for some plants to really persist and adapt to their environment. But if you were to rank out all the different pathogens of plants, fungi win. Compared to bacteria, compared to viruses, fungi rank top. There's more fungal species out there compared to bacteria and viruses that can cause problems. So this is a chart trying to show you the solubility under different pHs. So on the bottom of this chart, you see the pH range from 4 on the left to getting higher and higher as you work your way out. And then you can see wherever the bar is fatter, so nitrogen, wherever it's fatter, that's where it's more soluble. So here, phosphorus, you can see a big shift where it's soluble around 5.5. And then you can see as you go into alkaline conditions, 7.5, 8, phosphorus is just tanks. The plants will have a really hard time getting access to the phosphorus. And so that's how you interpret this chart. You see that again with manganese and boron, zinc, right? So wherever it's fattest, that's the pH range that it's most soluble and most available to the plant. This next diagram is the same thing. It's just color coded. So if that's better for your visual looking at it, you can use this one as well. But basically saying the same thing, slightly different resource. So if you're curious, why is that the case? Why is most soil, in general, we can say most soil is acidic. You can imagine rain coming down onto the soil, and then think about all the activities going on in the soil with the breakdown of organic matter. When microbes are breaking down organic matter, they are carrying out respiration. Many of them carry out respiration very similar to what we do. And so they give off CO2, just like we do. And again, once you react CO2 with water, you do get H+, which will drop, make your pH drop, get more acidic, right? So acidic soil is good in the sense that it allows for good solubility of many of the minerals that plants need. It can create some problems. For example, aluminum compounds can solubilize. Aluminum is toxic to plants. It's toxic to any organism in high enough amounts. So that's not necessarily a good thing. So having some metal contamination in your soils can be concerning. Iron, on the other hand, plants need iron. They need iron in fairly good amounts. So that can be a good thing within reason. But what can happen is aluminum, for example, can react with phosphate, and you'll get this compound that is basically insoluble. So if we say insoluble, that means it's precipitated out. It's not mixed into solution and it didn't dissolve. And so it's not such a big deal with the aluminum because plants don't really need a lot of aluminum anyway, so that's not the big deal. The big deal is the phosphorus. You complex phosphorus out and it's become insoluble. And that's a problem. And so when you sample your soil, let's say you want to grow plants in, you want to do some gardening, you should have your soil tested by a reputable company, and they should be able to tell you what kind of phosphorus you have. Do you have the readily available kind, or is it complexed out? And they can run tests to tell you that information. And so make sure that you understand that just because you might have a lot of phosphorus, you need to know if it's readily available or not to the plant. And so phosphorus deficiencies is a big problem with plants in general. So if you get too acidic, if your soil is too acidic and you've sent your sample off and they say, yep, you need to amend your soil, you need to raise it a little bit. It's not that hard to do. You can calcium, magnesium, so-called lime, you can add that in, calcium carbonate. So there's pretty easy ways to manipulate your pH if you have just a small garden. It's a little bit more challenging when you have acres and acres, but basically what you're trying to do is create a reaction that complexes the hydrogen ion or the proton. And if you can get that complex, then you've raised your pH because it's no longer free hydrogen ion. It's complex with something. So that will start to raise your pH to get closer to neutrality or more like just slightly acidic, not really acidic. So you can, that 5.5 to 6.5, if you start dropping too low, below 5.5, it can create some interesting problems but you can manipulate it back into the right range. So one of the ideas is that fungi like acidic soils. And so mycorrhizal fungi are incredibly important for plant growth. And I'm hoping you will learn that through your lab experiments that some of you will be conducting in lab. And I remember when I first learned about mycorrhizal fungi as an undergrad in my first plant phys class, I was just stunned. I mean, back then it was relatively unheard of. People just didn't talk about it very much because they didn't recognize the importance of this group of fungi. And I was just stunned to learn about it. And I fell in love with mycorrhizal fungi. I just thought, wow, this is amazing. These two very, very different organisms, and they will come together and form a relationship that's very unique and very much a cooperative relationship where they both win from that teaming up. And so we're looking at this idea of a fungus root symbiosis, and the fancy word is mycorrhizae. When you say that word or you see that word, that means you're talking about the roots that have formed that relationship. They're symbiotic with the fungus. So mycorrhizae. Anytime you see myco, that's referring to fungus. Anytime you see rhiza or rhizo, that's root. So fungus root is literally what that means. And experts say 80%, I've seen that push to 90%, depending on the expert, of plants will have this relationship with some type of mycorrhizal fungi. And in fact, evolutionary biologists state that mycorrhizal fungi were key to get plants out of the ocean and onto terrestrial systems. That the mycorrhizal fungi were there millions of years ago to help that process. And so really, the plants have the mycorrhizal fungi to thank for being able to adapt to dry land and deal with that. So here you're seeing an image of a comparison between two major groups of mycorrhizal fungi. This is just two. There are other groups. I don't have time to talk about all of them, but these two are very good to think about and compare. And so here you're seeing a plant root in each case in longitudinal view, and then the fungal hyphae you can see is coming off of the root system. And fungal hyphae, if you've ever looked at fungi, the fungal hyphae under a microscope, they're very fine in diameter. And we say they're filamentous or hair-like, all right? So that's what they're trying to show here. Compared, and you can just see here, compared to even the root hair, they are finer in diameter, okay? They're certainly finer than even the finest roots. They're even finer than the root hairs. So they're just much more petite in that regard, in their diameter. So keep that in mind. And then here you see there's two terms here. The arbuscular endomycorrhiza is what's being shown on the left, and then the ectomycorrhiza is what's being shown on the right. And we're going to get into these two groups and come back to some pictures. But basically, when they started realizing that so many of the plants that have been studied have this relationship, and they start thinking about well, there must be a benefit if they're forming this relationship and going through all this trouble to form a partnership. And so really what they are realizing and have realized is that the fungal hyphae that is associated with the roots really does expand the root system tremendously. And not just that, but it allows the roots to get into areas of the soil that the roots physically cannot make it into because they're too big. But the hyphae coming off the roots can penetrate into those nooks and crannies of the soil. And that would, again, just greatly expand the ability of the plant to get access to water and other minerals and things like that that it normally could never do. So let's look at a picture of that here. So here under the light microscope, you're seeing the roots are the dark thick images that you're seeing there. And then all of that finer web-like growth, that's all fungal hyphae. So again, looking at this visually, you can see how the fungal hyphae is much thinner in diameter compared to the root. So for the plant, that's really good because now it can get access to those parts of the soil that it never could before if it didn't have this relationship. actually just the opposite. So the question was like, if you have a really clay soil, would the fungus have a hard time? And actually they help the plant in heavy soils like that. They can actually help the plant because fungi have awesome enzymes and capabilities of kind of breaking down things around them and making room for themselves to continue to grow. So this is a good visual to keep in mind. So this chart is showing some of the types of mycorrhizal fungi. So you see here vesicular or arbuscular mycorrhizae. Sometimes they drop the vesicular. I'm seeing that more and more in more recent publications. They drop the vesicular and just say arbuscular mycorrhizae. So that group of fungi have, and then lots of different terms over the decades of research. Ectomycorrhiza are another type, but you see that there's others here. Endo, and monotropoid, those are really interesting. Orchidaceous, those are really bizarre groups. Varicoid again. So there's different groups out there that all have some very interesting attributes and different associations with different groups of plants. No. So the orchidaceous one, that's an interesting one because orchid seed, like there's a patch of woods I walk through all the time, and one year I saw over a dozen lady slippers. Just beautiful, beautiful walkway right along the trail. Beautiful lady slippers. And then the next year, nothing. And so if you've been hiking around where you see that pattern, what's happening is the orchid seeds that might have been created, at the end of the season, they need a very specific mycorrhizal fungus to germinate. Otherwise, the orchids don't even germinate. They won't germinate. It's weird. If you've ever looked at an orchid seed, it looks like a flake of dust floating, like particles you see floating around in sunshine. They're that small. It's weird. And so they absolutely need the right fungal partner, and it's very specific. And so if they can't meet up, I always wonder as I'm walking through the woods, and if you see a lady slipper, you should be in awe because not only are they beautiful, but what's the likelihood that this piece of dust seed and the absolute correct mycorrhizal fungus find each other in a natural setting? That's pretty, really interesting dynamic there. We're going to focus on the top two, the so-called vesicular arbuscular and ectomycorrhiza. I did my PhD work with ectomycorrhiza because back then we knew how to grow them in the lab. They were easy to work with, relatively. They grow slow, but at least you can grow them separate from the plant. The arbuscular mycorrhiza, you can't grow them separate from the plant. They're obligate. They have to hang out with the plant or they won't grow. And so working with them in the lab could be the tulips. Well, one of the things about the tulip industry is have you seen tulips that when the flowers are striped? That was a craze where they didn't know what caused that. And they actually referred to that period of time as tulip mania. And it's actually a virus that causes that streaking pattern. But back then they didn't even know about viruses. And so there's a book, I think it's at the library. I read through part of it. But it was actually like people would trade tulip bulbs, almost like you trade gold with each other. It was that desirable to have that streaky petal color. And so anyways, now they know how to engineer plants. Anyways, we're going to focus on these two. The ectomycorrhiza that I worked with, they hang out with a lot of trees here in the UP, of course, have ectomycorrhizal fungi, and we were working with red pine trees. So here, distinguishing features, it says fungi produce hyphae, arbuscles, and vesicles in plant root. And so that's a little abrupt. I just asterisk that to let you know that the fungal hyphae in the either the ecto or the VAM type mycorrhizal fungi, they don't actually get into the root cells. They're in the root, but not into the root cells. Okay, that is a very important distinction. Because if you're a plant and you're living your life and this fungus grows up to you and says, hey, let's form a partnership, you wouldn't want that fungus to actually penetrate into your cells. That's That's a boundary breach. Respect my boundaries, right? And so if a fungus does that, actually gets into your cytoplasm, starts, mucking around in there, that's going to trigger you to try to kill it, right? Because that's what pathogens do. They want to get into the cytoplasm where all the luscious nutrients are. So if a mycorrhizal fungus tries to do that, the plant will immediately kick into defense and kill. So every study that they've done, all the beautiful electron microscope studies that they've done, you can show that the plant will engineer a situation where there's always a membrane, its own membrane, it's usually the cell membrane, that is surrounding any part of the fungal hyphae, so that it's never, the hyphae isn't like swimming around and freely in the cytoplasm, right? So that's a big deal. It makes a point of the plant can then keep that relationship and not attack the fungus. So this is actually showing some of the electron microscope pictures where they talk about the TM is referring to the membrane that the plant is creating to surround any of the fungal hyphae to make sure that there's no free living hyphae. All right, so again, they're not going to pass through the membrane. All right, this is a big deal. The reason I'm making a big deal about it is I've seen students on the exam tell me that the fungal hyphae are in the plant root cells. If you state it that way, that means you think they're swimming around in the cytoplasm. So you have to be, details matter on how you phrase that. And the other reason why I'm making a point of this is so many different resources make it sound like the hyphae is swimming around in the cytoplasm. It drives me nuts. And so I'm also hoping you will educate other people that might be talking about mycorrhizal fungi, that is not the case. There's always a protective plant membrane surrounding the mycorrhizal hyphae. Okay, so the first group of mycorrhizal fungi that were capable of being more easily studied were the ectomycorrhizae because of their growing habitats. And so I'm going to talk about them first, but here in this visual, you can see a comparison how all of this black stuff that the artist drew, that's the fungal hyphae that is growing on the surface of the root. So that's why they're called ecto, because they grow very significantly on the root. And with the naked eye, if you can train yourself, like if you're out hiking around and you see a tree that got blown over and the root systems are visible, I always carry a hand lens with me because my boys, when they were little, they would just roll their eyes. Oh, look at these root system, and I try to get them to look at the mycorrhizal fungi, and they're just like, oh my god. But you can see them once you train your eye to it. You can see the ectomycorrhizal root system, and it's really cute. They look really delicate and cute. But so the word for that is down here in the image, mantle. So M-A-N-T-L-E, the fungal mantle. When someone says, oh, look at that fungal mantle, what they're referring to is this very heavy, dense growth of fungal tissue on the surface of the root. And it's so dense that it's almost like mushroom-like quality, like, the mushrooms you have on your pizza. It's like that dense. When you eat a mushroom, it's just a bunch of fungal hyphae, right? So it's just all really dense. So that's what it looks like. I'll show you some other pictures. The fruiting body that you see here, many of the mushrooms or fruiting bodies that you see when you're out hiking around near a tree, a lot of those are ectomycorrhizal fungi fruiting bodies. And that's their way of going through having sex, essentially. When they form that fruiting body, parts of the fruiting body are going through sexual reproduction. So it's their version of having sex. All right. So here you can see the fungal hyphae are penetrating into the root tissue. And what they do is they grow in between the root cells. So they're in the cell wall space. All those hyphae is like just going all around the cells in the cell wall space. The gentleman who discovered that, the scientist, his name was Hartig, Dr. Hartig, so they named it after him. And we call it the Hartig net. That's another term. So mantle, Hartig net. Okay. No, the Hartig net is what's growing inside. Oh. And so they call it a net because if you were to take the root tissue and dissolve it away, what's left, if you were just to imagine the fungal hyphae, it does look like a fishing net morphology. So they call it the Hartig net. So that thick mantle of fungal mycelium or hyphae that are growing on the roots, around the root, And then don't forget, there's also hyphae that will grow off the root, because that's a huge deal. The plant really wants that, so they have to grow off the root and into the soil. And then some of the hyphae will grow into the tissue, but it's just in the cell wall space. It's just the hyphae will kind of course through the tissue and grow in the cell wall space. So ectomycorrhizal, when you dig up a plant that has ectomycorrhizae, the biomass is significant. Okay, so often the total mass is comparable to the actual root biomass. And so that's a lot of fungal tissue. So it's very, very significant. Around here, we find them in most of the trees that you're looking at as you're walking across campus or in the woods, the shrubs, they have ectomycorrhizal partnerships. So if you're out hiking around and you see fruiting bodies at the base of a tree, chances are it's ectomycorrhizal. Okay. Chanterelle, anyone eaten chanterelle? Do you like it?
Yeah, I found some.
Yeah, they do. The apricot is very strong. I know some people don't like the texture of mushrooms, but you can always like blend it up and put it into soup or something like that if you don't like chewing on it. Yeah, they're usually on the ground. Well, blueberry, but again, sometimes you'll see them growing in a circle, the fairy ring idea. That is usually ectomycorrhizal. So if you are growing a plant in a rhizotron system, this is pressed up against plexiglass, and you inoculate this conifer with ectomycorrhizal fungi. And so the thicker tannish things, those are the plant roots. And then all of that white fluffy stuff that you see extending out, that's all fungal hyphae. So it's super extensive. And then if you imagine when you're out walking around in the woods, as you're walking, think about all of that underneath you. You have just a tremendous amount of fungal hyphae in a forest ecosystem. It's just mind-blowing when they do the calculations. No, it's just picking up some of the fungal mantle that a little bit.
Oh, is that the mushroom?
Well, yeah, the mushroom. And then I just put this here behind you around. You can actually, in a nursery situation where they're raising trees for oak planting, you often see a fruiting body forming eventually. Here is another drawing, just trying to show the mantle. Here they're coming into sheath. I put the mantle, the fungal mantle, and then the hunted net as the hyphae coming and growing in between the root cells. And then here in cross section, you can see again through the really dense growth of fungal hyphae to form the mantle. And then this is more of the net configuration you can see where it looks net-like as the hyphae grow into the tissue. The right side is what you might see if you go to an overblown tree and look at the roots. One of the big giveaways is look for finer roots that are in a Y-shaped pattern. That Y-shaped morphology really only occurs when the plant and the fungus have formed a relationship, the ectomycorrhizal relationship. So normally, fine roots just grow out, just straight. But for some reason, when the fungus and the plant root meet and they start chemically communicating with each other, it causes the root to go into a Y shape. And they think that's being done to increase the surface area for the fungus to grow onto it. And so you can see those with the naked eye. If you look closely under, and I'll show you some pictures, you can actually see that under the, even the dissecting scope. Here's another light microscope image where you're seeing all of that blue. That's all compressed hyphae, so that's the mantle around the root. And then all the blue between the cells, that's the net. So it's, there's lots of images that you can see in published papers where they show the mantle and the hyphae net inside the root tissue. If you look at like sections of the root system, so here you can clearly see the difference on the left. You can see how the roots have really thickened up. That's the fungal hyphae, the mantle, that are doing that. You can even see some Y shape here, very easy. Whereas non-mycorrhizal, they tend to be just very minimal root system in comparison, thinner. Here under the microscope, you're seeing some images. So they dug up this root system and washed off all the soil. And you can see some of the roots are obviously colonized. You can see the lighter color in this case of the fungal hyphae and that Y shape happening. And so the bottom right picture is with the naked eye. This is what you would see. That's the size for some of the ectomycorrhizal. If you're out in the woods and see an upturned root system, you should look for that kind of formation where you can see on some of the finer roots that very thick Y-shaped root. That's the fungal mantle. And then the other two images on this are just trying to get across the point that don't forget that they have the fungal hyphae going off into the soil, which is another really huge benefit for the plant, to have those fine hyphae going out and searching for things that the plant needs. Depending on the fungal species, you can get different colors to the root system. The mantle will look different. So here, this one is Ctenopathum, that's the genus Ctenopathum, and you can see how the tissue is very black, but tan perhaps, or reddish tan. It can be white. I've seen purple. It just depends. Here again, some more. And so most of these images are blown up, but the bottom right is more what you can see with the naked eye. That's the size you'd be looking for. Okay. So they are so the ectomycorrhiza, they named it that way because it's such a significant hyphal growth on the surface of the root, forming that mantle. It can be very substantial, but don't forget that they also control between the cells and the cell wall space. So some of the ones that are more famous in the scientific realm is this one, Lacaria bicolor. So there's been a ton of research on this one. This is one that I worked on with my PhD work. And it was the first one to be sequenced, DNA to be sequenced. People all over the world are kind of using it as a guinea pig, ectomycorrhizal fungi. It's just a good model organism because it grows pretty readily in the lab, although all mycorrhizal fungi are slow growing. Lower right, what you're seeing there is a Petri plate where someone put a chunk of the tissue on there and it starts to grow. And so fungi will grow from the point of origin where you place it out on the plate. It grows in that circular pattern. And Lacaria bicolor, two colors, and it tends to do that. It'll form this lovely purple color, especially if you give it the right nutrients. The hyphae turn purple and then tan as well. And so if you're working with fungi on plate cultures, it's important to remember that the most active hyphae is along the perimeter of the growth. So if someone said to me, hey, subculture, take some of that and get it onto a new plate, I would not pick the center of the fungus. Most likely the hyphae is dead by now. You work toward the perimeter of the growth, and that's where it's most active. So Lacaria is super common around the world. You can find it hanging out with all kinds of different, especially tree species and coniferous forests. We have a lot of Lacaria out in this area as well, and fairly easy to maintain in the lab. So that's probably why it was picked as a guinea pig, is its broad prevalence ecologically, and then also it's easy to grow, fairly easy anyway. And even the fruiting body can be bicolored, where you'll see purplish portions of the fruiting body. So it's a good way to identify it if you're mushroom hunting. So here again, when they form a relationship, they have that thick mantle, the Y-shaped pattern to the lateral roots. And so it's easy to see even just with the naked eye once you train your eye. Here's this is taken from a published paper where they're looking at Lacaria and its use with poplar trees. And here they're actually growing the poplar in tissue culture work, where they have the poplar seedling germinating and growing on the tissue culture. And then what they did was they spot inoculated the mycorrhizal fungi, and then they circled here that's where they're seeing the initiation of the symbiosis event. And so this has been heavily studied, looking at how do these two very different organisms work toward a partnership. And so there's a lot of chemical communication going on. The fungus is releasing chemicals to the roots. The plant will do that back to the fungus. They pretty much have to say to each other, hey, we're friends, let's hook up. All right. And so they have to be able to distinguish friend from foe, and then the right fungus to the correct plant. Sometimes it's a pretty tight, limited, only certain species can interact. Leucaria, on the other hand, has a pretty broad host range. It can hang out with a lot of different plants. Okay, so here you're seeing again the root system well colonized by Leucaria. It's sending off hyphae. Again, they're showing the fungal mantle and the growth in between the cells and cell wall space. There's even a paper that was done where they looked at Leucaria with certain pine trees, and the title, this wasn't the scientific title, but root fungi can turn pine trees into carnivores, which is, you know, pretty provocative wording, or at least encompasses. Root fungi may confer dark but useful powers on their plant hosts. So they had to get dramatic about it, but basically, you can read through this on your own, but basically what they found is that the fungus, or yeah, Leucaria was actually kind of preying on springtails, insects in the ground, and then digesting them and giving them the nutrients to the plant. And so it was, you know, it was like the Leucaria was hunting down the springtails. And so it was an interesting experiment that was done, and it kind of it was serendipitous how it led to this idea that Leucaria is actually essentially hunting down, in a way, the springtails, and then digesting, and then using that nutrition to help the plant. So the nitrogen in insects, there's a lot of nitrogen in the exoskeleton of insects. And so that was good. The plant needs a lot of nitrogen. So that was a very interesting interaction that initially when they set up that experiment, that's not what they were trying to even do. It was just kind of a serendipitous discovery. So less than 10% of the springtails placed of the experiment at the beginning were alive at the end. So they wiped out the springtails when they set up a controlled experiment. There's 47 wily survivors out of 500. So very interesting dynamics. So much more to learn when it comes to the mycorrhizal world. We're still in its infancy. So another one that I studied for my PhD is famous as well, Ctenococcum geophyllum, because again, huge broad host range. You can find it even growing up in the Arctic, and but it can be found in tropical areas. So it's a very adaptable fungus, and it grows slower than Leucaria for sure. It would take weeks for me to get enough tissue to work with. Beautiful tissue though on the agar plate. It forms this black velvet growth that's just beautiful. It's just really slow. So that's another one that's been heavily studied because it can form relationships with all kinds of different species, and it can live in different habitats. And one of the other reasons why I liked it is that it can release antibiotics that can kill plant pathogens. And I was very interested in biocontrol, still am. Here's some other species. Just trying to get your eye trained to what it looks like. I like that one because it's showing all the fine hyphae coming off. That's very important. Okay, so ectomycorrhizal fungi, a ton of fun. Next time you're out, maybe on the weekend, look for an upturned tree, bring a hand lens or good glasses or something. See if you can find some ectomycorrhizal fungi. All right, we'll get into the endomycorrhizal fungi on Monday.